Welcome back. This is the first lecture of Chapter 10, and Chapter 10 is going to be the last unit, or the last chapter of the first semester. And so we're going to talk about all the components of Chapter 10, all the material in Chapter 10 in about 4 lectures. This first lecture is going to be kind of a review of the cell theory and an intro, kind of a brief intro into what cell division is and what it's used for. In later videos, we're going to talk about the specifics of mitosis. We're going to talk about the specifics of cytokinesis. We're also going to talk about the specifics of binary fission, what the cell cycle is and how the cell cycle is regulated, and what happens if that cell cycle becomes unregulated, specifically the production of cancer cells and how those cells produce tumors and that kind of thing. So I don't foresee this video taking very long, and so we'll go ahead and just begin. Remember when we talked about the cell theory in Chapter 7, there were 3 components. The first component is all living things are composed of cells. The second component is cells are the basic unit. Cells are the basic unit of structure and function in living things. And the third component is cells are produced from pre-existing cells. And so I bring that up because now we're going to dive into what that third component really means. Remember that it was Rudolf Virchow. And Rudolf Virchow talked about, after studying mitosis, the importance of this pre-existing cell idea, meaning spontaneous generation needs to be disproven, and he wasn't the only one that contributed to disproving spontaneous generation, but he was the one that was very influential in disproving that because he studied mitosis. He was the one that watched cells divide, and when you watch cells divide, you know that, or you notice, you make an observation that the new cells, the 2 new cells, not only came from the parent cell, but that the 2 daughter cells are identical genetically, or should be identical genetically, to that one parent cell. Okay, and so he did some work in 1858 and contributed that third component to the cell theory. And remember, that third component is that cells are produced from pre-existing cells, or cells arrive from pre-existing cells. Okay, cell division is commonly called mitosis, but I want to make a distinction right now, and I want to make it very clear that cell division, while a lot of times referred to as mitosis, and while mitosis is referred to a lot as cell division, those are entirely different ideas. Okay, cell division is specifically dividing the cell, which is more synonymous with cytokinesis. And again, we're going to talk about what cytokinesis is. Cell division means splitting the cytoplasm so that you produce 2 genetically identical cells. Mitosis is specifically the division of the nucleus. Okay, so mitosis is a process in which the chromosomes are going to be duplicated essentially and then split evenly. Well, cells can divide the nucleus and not divide the cell. Plant cells do it all the time, meaning one nucleus within one cell is split, and the cytoplasm is never split between those cells. Okay, that happens all the time. Mitosis would happen, but cell division didn't happen. And so mitosis is the division of the nucleus. Cell division would be more synonymous with cytokinesis, which is division of the cytoplasm. Okay, there are technically 3 reasons for a cell to divide. The 2 big reasons are here, and the third reason, which isn't on here, would be growth. Okay, and we'll talk about what each of these 3 mean. So cells will go through cell division for 3 reasons. 1, sometimes cells die, and sometimes cells are damaged. An example would be you go out to the beach or you go out to the lake and don't apply sunscreen as often or at all, and you end up sunburned. Okay, those cells are damaged, and in a lot of cases they die. That's probably not hard for you to believe, because a lot of times when you get a sunburn, especially if the sunburn's bad, the skin ends up flaking off or ends up peeling. Okay, that's a pretty good indication that the cells have died. And it's good for us as an organism that cells can replace dead and damaged cells, so that you don't have to live with the burnt cells or the damaged cells or the dead cells for the rest of your life. Okay, if cells didn't replace dead and damaged cells, then the first sunburn you ever experience would be essentially irreversible, meaning you would have to live with the damage that was done to those cells for the rest of your life. Okay, for instance, or another example, if you decide to take a pan of cookies out of a oven using a wet paper towel, you'll burn your fingers, and the damage that is done to your fingertips will be not irreversible. You will fix the cells that are making up. You'll fix the cells making up the skin of your fingertips, and you won't have to live with burnt fingertips for the rest of your life. Okay, so replace dead and damaged cells. Another one, ensure genetic continuity. This is a really important concept. What does that mean? It means that you are making sure or ensuring that the cells that are made from the last generation are the exact same genetically as they are now. Okay, when a sperm cell and egg cell were joined during conception, those two kind of half cells, if you will, and I know we haven't gone through genetics yet, and we haven't talked about diploid cells and what that means, but essentially sperm cells and egg cells are half cells, or half nuclei. They contain a half nuclei, and when they fuse, they produce one cell that has a full nuclei. And so you obviously are made up of several trillion cells now, but you started with one cell. How does that one cell get to the several trillion cells that make you now, or make you up now? And the answer is cell division. Okay, the one cell divides and becomes 2, and the 2 divide and become 4, and the 4 divide and become 8, and the 8 divide and become 16. And you can see that every time you're doubling the number, and so it takes a while to get a substantial number, but once you kind of increase the amount of cells, it takes very little time. In fact, it takes 9 months to produce a baby that is made up of tons and tons of cells that are all genetically identical, or should be genetically identical, to the first cell that made up that zygote. Okay, and then growth. And I kind of just talked about it, how one zygote, or a combination of sperm and egg cell, basically grow into a baby. And then once the baby's born, the growth doesn't stop. The growth continues into adulthood, where you increase the number of cells. And it's one good distinction to make, that an adult doesn't have bigger cells than a baby. In fact, baby cells and adult cells are identical in terms of size. The difference is in the number of cells. Okay, and so if an organism needs to grow, or wants to grow, or has to grow, it grows not by the size of cell, it grows by the number of cells that it contains. And how does it grow by the number? The number that it currently has to divide and produce bigger numbers. Okay, so why does a cell remain small? And we talked about it when we did chapter 7, but large cells, remember, are inefficient. And when we talk about inefficiency, remember we talked about gas exchange, we talked about waste removal, we talked about food, or bringing food in, bringing water in, bringing waste out, bringing O2 in and CO2 out, and we talked about surface area to volume ratio. And remember, we basically we determined that small is better. Why is it better? If you are a small cell, meaning if you're a cell this big versus a cell that's this big, and you need to get materials from the outside to the very middle of a cell, you're going to take essentially twice as long to get your materials into there as you do to get your materials in here. If you were to measure these two lines, this is basically 1/2 x, if you want to put a variable on it, and this would be x. Okay? It's easier, and it takes half as much time to get food to that point. It takes half as much time in these two cases to get O2 to that point. It takes half as much time to get CO2 out and waste products out from that point. Okay, and so I don't want to hash out a lot of things because we've already talked about this, but just as a review, surface area to volume ratio is still definitely a player in this unit, and it's one to think about going forward. One of the other reasons why cells remain small or need to remain small is this idea of DNA overload. And what does that mean? We will talk about DNA in a lot more detail in Chapter 12 next semester, but basically DNA is the set of instructions for all cell activities. Okay, and so we've talked about the nucleus being the control center of the cell. Well, it's the control center of the cell because it contains all the DNA, and the DNA is basically the roadmap for the cell, or the overall set of instructions for that cell. There is only, or should only be, one set of DNA per cell, and every cell in your body should be or contain the exact same DNA. So what makes a liver cell different than a skin cell? Because the liver cell and skin cell have the exact same DNA. And so the difference between a skin cell and a liver cell is which genes are turned on and which genes are turned off. A liver cell contains a huge amount of DNA, but it turns on the liver genes, and so it basically lives as a liver cell. Skin cells contain liver genes, and they also contain skin cell genes, but they turn the skin cell genes on and the liver genes off. And so every single cell in your body contains the same set of DNA. It just depends on which genes are turned on and which genes are turned off. And we're going to talk about genetics later, but just as a kind of a precursor to Chapter 12. Sorry about that. But like I was saying, just as a precursor to Chapter 12, certain cells have genes that are turned on and certain genes turned off. And so smaller is better. And this is an image that I'm going to leave you. Remember, we talked about surface area to volume ratio. We know that if we have a small cube that is 1 by 1 by 1, it is going to give you 6 total surface area units, and the volume is going to be 1. And so you get a 6 to 1 or a 6 surface area to volume ratio. If you make this basically a lot bigger, you get 150 surface area to 125. That's a 1.2 to 1. And so you can see the difference between these two. But that's where we're going to end it. Until next time, see ya.